Hello students, the topic of today's module is thermogenesis. Thermogenesis can be literally defined as production of heat in organisms. It is an important physiological process as well as a normal byproduct of metabolic processes. Increased thermogenesis can be observed following injury, inflammation, infection, physical or emotional stress, and in certain chronic diseases such as malignancy. Thermogenesis is also a primary effector of thermoregulation in homeotherms and an important mediator of fever. There are three types of thermogenesis: shivering thermogenesis, dietary thermogenesis, and non-shivering thermogenesis or adaptive thermogenesis. Shivering thermogenesis occurs when practically all of the skeletal muscle groups are involuntarily driven to contract synchronously. Heat production during cold exposure can be increased by nearly 3 to 4-fold by shivering. Next, dietary thermogenesis, also called the specific dynamic effect of food or meal-induced thermogenesis, refers to the heat produced in response to and following the consumption of a meal. The ingestion of nutrients causes an obligatory increase in heat production by the body as a result of the metabolic costs of digestion, absorption, metabolism, and storage of nutrients. This heat is not useful to an animal that is living in a thermal neutral environment, but will contribute to the maintenance of body temperature when an animal is exposed to a cold environment. Then, non-shivering thermogenesis is defined as a heat produced from metabolic processes not involving contracting muscles. So, apart from thermogenesis through muscle activity, heat can be produced by non-shivering thermogenesis. This heat production occurs in brown adipose tissue or BAT. In this module, we will be focusing on the third type of thermogenesis, that is non-shivering or adaptive thermogenesis. A key strategy for mammals subjected to chronic cold exposure is to either reduce heat loss by adding insulation or accelerate oxygen consumption and produce heat, but without muscular contraction. The former mechanism is not a viable option in relatively small mammals that are limited in the amount of insulative fur that can be added from summer to winter. Smaller species of mammals must either develop better thermogenic capacity in the winter, which also requires more foraging for food, or they hibernate to avoid the metabolic costs of thermoregulation in the winter. The heat produced from shivering is effective at counteracting the heat loss from cold exposure, but shivering is metabolically inefficient and uncomfortable. Ideally, non-shivering or adaptive thermogenesis is the most effective way to adapt to a cold environment. Some organisms possess the ability to uncouple oxidative phosphorylation from ATP synthesis in mitochondria to generate heat. Such uncoupling is a means to maintain body temperature in hibernating animals, in some newborn animals, including humans, and in mammals adapted to very cold environments. In hibernating animals, in some newborn animals, including humans, and in mammals adapted to cold, brown fat, which is brown adipose tissue, is specialized tissue for this process of non-shivering thermogenesis. Brown adipose tissue is very rich in mitochondria, often called brown fat mitochondria. The tissue appears brown from the combination of the greenish-colored cytochromes in the numerous mitochondria and the red hemoglobin present in the extensive blood supply, which helps to carry the heat throughout the body. So let's see how uncoupling of electron transport takes place from ATP synthesis. To understand the molecular basis of thermogenic activity, it is necessary to remember the fundamentals of mitochondrial bioenergetics. Mitochondria are the cellular organelles where respiration occurs and where cellular energy is produced in the form of ATP. They contain two compartments bounded by inner and outer membranes. The outer membrane is permeable to small metabolites, whereas the permeability of the inner membrane is controlled to maintain the high electrochemical gradient created by the mitochondrial respiratory chain that is necessary for energy conservation and ATP synthesis in mitochondria. The inner membrane transports anion substrates such as ADP, ATP, phosphates, oxoglutarate, citrates, glutamate, and malate. The reactions of the citric acid cycle, fatty acid oxidation, and several steps of urea synthesis and gluconeogenesis also take place in mitochondria. Energy produced by mitochondrial respiration is used for ATP synthesis by a complex mechanism referred to as oxidative phosphorylation. NADH and FADH2 are energy-rich molecules which are formed in glycolysis, fatty acid oxidation, and the citric acid cycle. So, in oxidative phosphorylation, the transfer of the electrons from NADH or FADH2 to molecular oxygen by a series of electron carriers in the electron transport chain leads to the pumping out of the protons out of the mitochondrial matrix. The resulting electrochemical proton gradient due to this proton pumping is harnessed to make ATP by ATP synthase, which is another transmembrane protein complex present in the inner mitochondrial membrane. In addition to oxidative phosphorylation and metabolic pathways, mitochondria are involved in thermogenesis. Let's see how. Peter Mitchell's chemiosmotic theory demonstrated that the mitochondrial electrochemical proton gradient generated as electrons are passed down the respiratory chain. It is the primary source for cellular ATP synthesis. The mitochondrial respiratory chain is made of 5 complexes. Complex 1, 3, and 4 pump protons outside the inner membrane during reoxidation of coenzymes and generate a proton gradient that is consumed by complex 5 or ATP synthase, which catalyzes ATP synthesis. In addition to re-entry of protons through ATP synthase, a proton leak represents another mechanism consuming the mitochondrial proton gradient. Mitchell's theory predicted that any proton leak not coupled with ATP synthesis would provoke uncoupling of respiration and thermogenesis. So, the tight coupling of electron transport and phosphorylation in mitochondria can be uncoupled by certain substances which we call uncouplers. They may be chemical or physiological uncouplers. Such an uncoupling of respiration to ADP phosphorylation is represented by the mitochondrial uncoupling protein of brown adipocytes, which dissipates energy of the substrate oxidation as heat. Thermogenesis in brown adipose tissue or BAT is achieved because uncoupling protein UCP1 allows the re-entry of protons into the matrix and therefore uncouples respiration from ATP synthesis. Besides adaptive thermogenesis, uncoupling of respiration allows continuous reoxidation of coenzymes that are essential to metabolic pathways. In fact, partial uncoupling of respiration prevents an exaggerated increase in ATP level that would inhibit respiration altogether. UCP1 is a mitochondrial membrane transporter essential to non-shivering thermogenesis and control of body temperature. UCP1 is also known as thermogenin, which is a transmembrane protein dimer that resembles an ATP-ADP translocator. It is expressed in high levels in brown adipocytes, where it may account for up to 4% of the total protein and 8% of the mitochondrial protein. UCP1 forms a pathway for the flow of protons from the cytoplasm to the matrix. In essence, UCP1 generates heat by short-circuiting the mitochondrial proton battery. The energy of the proton gradient, normally captured as ATP, is released as heat as the protons flow through UCP1 to the mitochondrial matrix. This dissipative proton pathway is activated when the core body temperature begins to fall. In response to a temperature drop, the release of the hormones leads to liberation of free fatty acids from triacylglycerols that in turn activate thermogenin. In addition to UCP1, two other uncoupling proteins have been identified. UCP2, which is 56% identical in sequence with UCP1, is found in a wide variety of tissues. UCP2 is not involved in cold-induced thermogenesis, but may be involved in the resting metabolic rate of the animal. UCP3, which is 57% identical with UCP1 and 73% identical with UCP2, is localized to skeletal muscle and brown fat. UCP3 is not involved in cold-induced thermogenesis. This family of uncoupling proteins, especially UCP2 and UCP3, may play a role in energy homeostasis. It was also suggested that they regulate fatty acid metabolism by exporting fatty acid anions outside of the mitochondrial matrix when a large excess of fatty acids is inside the mitochondria. The role of the plant uncoupling proteins is still being elucidated. Some of them, which are proteins from potato, Arabidopsis, and the cabbage, have been shown to respond when plants are subjected to cold stress, suggesting that they have a thermogenic role analogous to that of mammalian UCP1. Specific plant functions also involve UCP-mediated energy dissipation. During fruit ripening, heat is generated, and it had been previously thought that the thermogenesis was mainly due to the induction in the respiratory chain of the alternative oxidase. However, it has already been demonstrated in tomato and mango that UCPs play an essential role in this process. The discovery of UCPs in many different eukaryotic organisms suggests that the regulation of energetic efficiency through the physiological uncoupling of oxidative phosphorylation may be a common strategy developed early in evolution. So that's all we'll be learning today. Let us summarize the main points before we conclude. Thermogenesis is the generation of heat in organisms and a very important physiological process. There are three types of thermogenesis: shivering thermogenesis, dietary thermogenesis, and non-shivering or adaptive thermogenesis. Non-shivering thermogenesis is observed in organisms that possess the ability to uncouple oxidative phosphorylation from ATP synthesis to generate body heat. Such uncoupling is a means to maintain body temperatures in hibernating animals, in some newborn animals, including humans, and in mammals, which are adapted to very cold environments. In mitochondrial oxidative phosphorylation, the transfer of electrons from NADH or FADH2 to molecular oxygen by a series of electron carriers in the electron transport chain leads to pumping out of the protons out of the mitochondrial matrix. The resulting electrochemical proton gradient due to this proton pumping is harnessed to make ATP by ATP synthase. Uncoupling proteins like UCP1, allow the protons to leak into the mitochondrial matrix, thus dissipating the proton motive force across the inner mitochondrial membrane. Phosphorylation by ATP synthase fails, due to which ATP is not synthesized and the energy is released as heat. This is the basis of adaptive thermogenesis, which allows organisms to survive in extreme cold temperatures. Thank you.